All right, so now let's get on to replication. Replication is actually fairly complicated. I don't expect that you know it in super duper detail, but you need to know some of the basics of it, okay? So during replication, there are some important proteins that are kind of like the key role players, if you would think of this as a play, okay? So the first one is called DNA helicase, and that one's important because it is going to be responsible for untwisting that DNA, so it's all coiled up. It's going to untwist it so that it's in that ladder shape, and then it's going to break apart those complementary strands into separate strands, okay? So it does that by breaking the hydrogen bonds. So the DNA helicase unwinds the helix of DNA. Okay, so you can think of it almost like unzipping that winding staircase. It just opens it up like a zipper. And I know this picture is kind of fuzzy, but I liked it because of the different colors. You can see here, this triangle is indicating that DNA helicase, okay? At the replication fork, the place where the strands are separated is called a replication fork. So that would be right here. This is our replication fork. And at that replication fork, there's going to be another protein called DNA polymerase, okay? And this is signified as this block structure right here. So the DNA polymerase is going to move along every strand and add new nucleotides to these exposed nitrogen bases. So look here. This is one of our strands, and the DNA polymerase is going to come in and add nucleotides to those open structures, okay? This process is going to keep going until all the DNA has been copied. The polymerase has been signaled to stop. It reaches like a signal in the DNA. It's like a little code that says stop right here. It's going to reach that signal, and then it's going to come off or detach. In eukaryotic cells, the DNA replication occurs at hundreds of locations at the same time, and it goes in both directions of the DNA until the entire chromosome is copied. And I told you that there's 46 chromosomes in every human cell. So this is happening at hundreds of locations on one chromosome, and it's being repeated on every other chromosome, and it typically happens all at the same time. Crazy that this is happening in your cells right now. Okay, so this is a little bit of a simplified picture of it, but here we have one of the complementary strands, and here's the other complementary strand. We have here is the replication fork right here. Okay, so that DNA helicase would be right in there, opening it up or unzipping that structure, and then the polymerase is going to come in right here, and it's going to start adding new nucleotides to this exposed strand of nucleotides right there. Okay. Here's a little video, and I wanted to show you kind of so you can get a picture of what I'm talking about, and then I'm going to talk about leading strands and lagging strands. So check this out.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces.
These orange structures right here are the DNA polymerase. This green one is the helicase.
First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Okay, so here are some things that I really wanted you to know. That will get a little bit more into detail than what you need to be able to talk about with me, but I do want you to understand the leading strand and the lagging strand and why it works that way, okay? So the first thing that you need to know is that those two strands of DNA that end up winding together are anti-parallel, and what that means is that sugar phosphate backbone is going to run in opposite directions, and Crick was the one that figured this one out, okay? So one strand runs in a 5' to 3' direction, while the other one runs in a 3' to 5'. And so I want to explain to you what that means. So if you look here, every carbon or every sugar is numbered. The carbons in that structure are numbered, okay? And you don't need to know how or why, but I'm explaining to you why there's a 5' end and a 3' end, okay? So if you look here, there's an oxygen here, and so we're going to count around clockwise. So 1, 2, 3, 4, and there's actually another sugar or another carbon right there. So that's 5, okay? So on this one, the 5 carbon is actually pointing up, okay? So that's the 5' end. If you look over here, this one is the 3' end because that 1, 2, 3 sugar is the one that's closest to this side and pointing up. Or sorry, that third carbon in the sugar is pointing up. Okay, so that's why it's called 5' and 3'. I don't expect you to know that. I'm just explaining to you so you understand why they're numbered that way. Okay, what you do need to know though is that because of the way that those are structured, the way that they're copied is going to be a little bit different. So DNA polymerase can only add nucleotides at this end of the DNA strand, okay, forming new strands. So it reads it from the 5' to the 3' direction. So on this side, it would end up going like this. The reason that that's important, and when I say that they're anti-parallel, I want you to see here. Sorry, I'm going to zoom back in a second. The phosphate group is on the 5' end, so on this side it's sticking up right here. And on this side, on this nucleotide, it's pointing down, okay? So parallel means, is like this, okay? They're not actually running parallel, they're running anti-parallel, okay? They're running backwards. They're formed backwards from each other, and that's the way that DNA is structured, and it does that on purpose. So because of that, we have what we call a leading strand and a lagging strand. If you can see on this picture, the leading strand is the one where the DNA polymerase adds nucleotides going toward the replication fork. So this is the one that's reading it in a 5'. It's adding from a 5' to a 3' direction. So this light blue strand right here is the one that's being added onto it. It can only add on to the 3' end, which is the one that's facing the replication fork, okay? The lagging strand, because of the way it's formed, has to be added to in a direction that goes away from the replication fork, okay? And the person that actually found this was a guy named Okazaki, and so we call those Okazaki fragments. So if you see here, there was, this is one little chunk, and then as the replication fork continued to open, then the polymerase jumped back here and replicated this way, and there's a second chunk or Okazaki fragment, and then once it got to here, it was able to jump back to where the new section of the replication fork was and copy this way again, okay, because it has to copy and it has to add on to the 3' end, okay? So the problem with that is, though, now we have all these chunks of DNA, and you do not want chunky DNA. That would be a major problem. So there is another protein called DNA ligase, and basically it's like a stapler. So it comes along and it adds those Okazaki fragments or chunks together so that it's one continuous strand of DNA, okay? The reason that we call that the lagging strand is because this side copies a little bit slower because it has to wait for that 3' end to open up at the replicating fork. So this side, the leading strand, can go in a continuous, ongoing fashion, where the lagging strand has to pretty much, it looks like, if you guys ever played Leapfrog, that's what it looks like. So it copies this way, and then it jumps back, and it copies this way, and then it jumps back and copies this way, and then it jumps back, and it keeps doing that until it gets to the very end of the DNA strand. All right, so let's practice really fast. You have your original strand, so of the DNA strand, we open it up, and on one of these strands has the bases C, A, T, G, T, G, C. What would be the copy of the new strand? What would those nucleotides be? Go ahead and write that down in your notes. Okay, got it? So my question to you is, what is the final product of replication? We'll talk about that question in class. I do have a few other questions for you that I want you to think about because I want to talk about these in class too. These are just recall questions, so see if you can answer these without glancing back in your notes. What is the site of replication called? How many strands of DNA are replicated at a time? And then I want you to make an analogy. What can you compare DNA replication to? Something that is like a common everyday human thing that you would come across. What could you compare DNA replication to? Just food for thought. All right, so why is this so important? Why do we need to know this? Well, we need to know the sequence of bases, and scientists can actually go in and determine what those sequence of bases are, and that's how they do like forensic DNA, cool CSI kind of stuff. But the strands of DNA are very unique in terms of they all have the same structure, they all have the same 4 bases, but the way those bases are sequenced is very unique for every person, okay? Even identical twins are gonna have somewhat slight differences, okay? My twins are a boy and a girl. They have very different DNA sequences. They're related, so they have similarities, but they have their own unique strands, okay? Well, those are so important because that DNA structure, okay, are the genes that are going to determine how the proteins in your body are going to be made. And remember, we talked about proteins make up so much of your body, okay? So a gene is a big section or a big chunk of DNA that makes up a single protein, okay? And every gene is going to be unique in the protein that it makes, okay? Proteins are important, and this is just a very short list of what proteins are, but remember that they're body structure, so that means they make up your muscle, they make up your bone, they make up your hair and your skin and the cartilage and the tendons, and all of the stuff that you see that makes up the structure of a person, that makes up you, is made out of proteins. Okay, they also act as enzymes. So enzymes are special proteins that speed up chemical reactions. So when we talked about DNA polymerase and DNA helicase and ligase, those are all enzymes that are helping these procedures happen. They're helping these chemical reactions happen. Okay, every chemical reaction has enzymes that help it go along. Otherwise, it would take too long and they would be too slow and it would take up too much energy. So these enzymes are protein helpers. Okay. Another important thing that proteins do for our bodies is they fight disease, okay? There are proteins that make up antibodies that help you fight off all the different infections that you come in contact with on a daily basis. Another important thing that they do, there's a protein that carries oxygen through your blood. That protein is called hemoglobin, and that protein is very specific in that it picks up the oxygen to take to all of the different parts of your body, so cell respiration can happen. And then as the oxygen diffuses out of the cell, the hemoglobin protein picks up the carbon dioxide and it takes it back to your lungs so you can get rid of it, okay? Please remember, proteins do not provide energy. They help with chemical reactions, but they are not a source of energy for your body. Your body goes to fats and carbohydrates for energy, okay? So just food for thought, what kind of foods provide proteins? I want you to think about that for just a few minutes, and that's also something we'll talk about in class. So what's the point of all of this? Why does DNA have to be copied? Who cares, right? Well, you should care. First of all, because we're going to talk about the cell cycle in a little bit, and you'll need to know when the section of DNA synthesis occurs. So that's one reason that you need to care. But the other reason that you need to care is because your cells are growing and dividing all the time, either because you're growing or because they're recycling and replacing old cells that aren't able to do their jobs anymore. Well, if all of your cells died without being copied, you would be gone. So your cells have a very important function. Before they die, we hope that they grow bigger and divide and grow bigger and divide and grow bigger and divide. So these cells are constantly recycling in your body, okay? So when cells divide, they have to have 2 identical copies of DNA in their nucleus, okay? One for each new cell. So what happens in the cell cycle, just a brief overview, is the cell gets bigger and does all of its normal cell functions. And then once it's decided it's gotten too big, then it's going to send a signal that the DNA starts copying. That DNA makes 2 identical copies. Okay, so you have 2 copies of 46 chromosomes in a cell in a nucleus, and then the cell's going to divide. So the chromosomes will all line up and they'll pull apart, and then the cells will pull apart too, so that each new cell has an identical copy of DNA. You want your cells to be identical. If they're not, that's a problem. Okay, so this is important for you to know because DNA is copied during this phase called interphase. And this whole part of the cell cycle is interphase, but this S phase right here, and we'll talk more about this when we talk about cell cycle, but that S phase is actually for synthesis, and that's when the DNA copies are being made. Okay, if this didn't happen before the cells divided, then the cell would divide that DNA unevenly and your cells would not be identical, and that would be a problem for your cells, okay? So there has to be an identical copy of DNA in every new cell. All right, so there you have it. That was DNA structure and replication. I am sure that you have questions. I'll be happy to answer those for you in class. And in the meantime, have a great day.